Welcome back to the third lecture in this part of the series of PS121. Today, the part of the lecture that I'm going to talk to you about is what are we doing for current or recent molecular genetic testing, and where are we going to dissect out the vast genetic heterogeneity of much, much rarer diseases than Duchenne muscular dystrophy, and how does this look in practice in clinic? What I'm showing here is a multiplexed PCR reaction for Duchenne muscular dystrophy or Becker muscular dystrophy diagnosis. And this is incredibly useful for deletion or duplication testing. And this is typically it's typically performed in clinical laboratories as a technique called multiplex ligation-dependent PCR analysis, also known as MLPA, which is simply a term to say I can run multiple different independent PCR fragments. They're running on the same gel because they're different sizes. Each size tells me exactly which exon is being looked at. So in this example, E is equal to exon 45. D is equal to exon 44. And if those are missing, so if I had an individual with E and F missing, that would be predictive of an exon 45 is deleted and an exon 48 is deleted, and you'd reason that 46 and 47 are also deleted. But you'd look for how big that deletion is, and this testing is actually quite accurate and quite useful and quite inexpensive. But we're moving towards a new era where we won't necessarily do all of our DNA diagnostics in a gene-specific fashion. So I'm going to walk you through what that looks like and why and how we're able to do this. And I attribute some of this to the success of identifying the DMD gene and the success of wanting to figure out the many thousands of forms of rare genetic diseases that affect humans. So Duchenne muscular dystrophy is at the very common end, and at the other end, so we probably have about 300,000 people affected by Duchenne living on the planet at any one instant. And at the far opposite end, we've identified human disease mutations that have occurred and have been identified in only one person, but we've reliably identified that that human disease gene occurs. We want to fill out that complete spectrum of ultra-rare disease to relatively common genetic diseases. We've done a lot of the work on the relatively common genetic diseases like Duchenne muscular dystrophy, but sometimes they can still masquerade or look like other various rare diseases. So we want generic tools that look at the whole genome very sensitively early in life and early on as a given clinical disease is manifesting. One of the ways that this has gotten to be possible is with the dramatically decreasing cost of generating raw and very high quality randomly sampled sequence from the human genome. The human genome, each genome from your mother and your father is about 3 billion letters, so total your genome is 6 billion letters of G, A, T's, and C's, and decoding those was the project of the Human Genome Project, which is planned to be about a 15-year project, and that was completed largely in about 2001. If you think about how much it would cost to then move forward doing sequencing, if we projected at that instant, if we wanted to sequence the second human genome, it would cost about $100 million at that time. Right now, it's possible to generate a full human genome sequence for about 4,000 or even less as new technology is moving forward. So we've moved into a realm which we now call next generation sequencing that allows us in a shotgun-based approach to sample the entire human genome from a given human and accurately reconstruct what their diploid genome looks like. If you think about this in historical perspective, it's really pretty dramatic, and it's happened over an incredibly short time period. So I was involved when I was doing my postdoc, developing technologies in relation to how do we search through and how do we find DNA mutations more efficiently. And the idea that we'd sequence the whole human genome was being actively debated. Was this a worthwhile enterprise? How long would it take? It ended up taking about 10 years of concerted effort, and cost billions of dollars to do, and gave us a reference map, which became profoundly important for figuring out many, many hundreds and hundreds of disease genes and giving us a roadmap of what genes there are that make up humans. Profoundly important. It didn't necessarily give us exactly the tools that would allow us to explore human genetic variation on individuals day in and day out. So in looking back in retrospect, most of the 24 billion bases of genomic sequence that were generated for the Human Genome Project for building the first reference map of the human genome were generated in 1 year by the Human Genome Project. And that cost $100 million or more in that final year of the project. And if you move forward, you could probably still do this with what we consider somewhat old school sequencing, Sanger sequencing at this point, for tens of millions of dollars. The first massively parallel genome that was sequenced was actually Jim Watson's genome was sequenced using the first next generation strategy, and that cost $2 million to do and was heralded as an amazing new way to move forward. We can now, in one laboratory, with one technician, with one machine, generate 10 times more data than was ever generated by the Human Genome Project per day. So the world is just so dramatically different that we can use these tools in very dramatically different ways, and indeed probe and look for de novo mutations genome-wide in humans in real clinical practice. So the way that this has been implemented is a bit of a shortcut or a bit of a simplification of the genome, just to try to expend and use our resources as best as possible. So the human exome is defined as that portion of the human genome that encodes all the exons. So exons are the parts of the gene that are glued together to make the full transcript that then are read into protein sequences. And it turns out that humans make about 20,000 reliable protein coding genes, and these are encoded in 240,000 exons. So we can develop, and multiple companies have now developed, very reliable mechanisms to capture and just pull out those 240,000 exons. So every single gene and every single protein coding base of genes are pulled down simultaneously. This represents about 1% of the human genome, or about 30 million bases, and we can oversample this 100 times over so that the likelihood of seeing the maternal allele is, we're seeing it 50 times over on average, and the likelihood of observing the paternal allele at a given base, we're also observing that paternal allele 50 times over. Because we're oversampling so much, we get incredibly accurate data, a very low likelihood that we've made mistakes in this type of data. And we can now look genome-wide for all protein coding bases, which is a spectacular tool, and we can now use it for decoding very rare undiagnosed people. So in 2012, UCLA, in a process that I was helpful in starting, created the Clinical Exome Sequencing Laboratory and the Clinical Genomics Center. And this process implemented in January of 2012 for the first time, a true clinical process where physicians anywhere in the world could order up clinical testing and genetic testing of their patients who they weren't able to diagnose, but they suspect have a genetic basis for the disease. And with that, the physician fills out a typical requisition, sends us a blood sample. Oftentimes we ask for a blood sample of the mother, the father, and the affected child or the affected individual, because oftentimes the mutation is identified as a de novo mutation, exactly like Duchenne muscular dystrophy, where I told you a third of all the patients are due to de novo mutations. And that's a powerful part of the diagnostic process for how we can figure out whether a given mutation is causal or not. The DNA sample is made from the blood, and then we go through a very consistent process that looks the same on every patient, and we've now done over 3,000 exomes in this process to try to identify and deliver back diagnostic information. A typical output from this clinical exome sequencing identifies over 20,000 DNA variants per person. And our goal is not to report back to a given physician or a family or an affected individual 20,000 DNA variants, because nobody would be able to make sense of it in any clear way. Instead, we filter all that data to try to come up with the one single base, the one single change that's causal for that child's genetic condition. So we're looking under a model where we're really only considering Mendelian genetic causes, but there's over 4,000 different Mendelian genetic conditions that lead to various human diseases, including a very large array of different genes that are involved in many different forms of muscular dystrophy. We looked at this process after we'd done our first 814 cases, and within that sampling from 142 different physicians at 42 different institutions, we were able to give back a specific molecular diagnosis and a specific molecular test for a given individual family 26% of the time across a wide spectrum of different diseases: muscular dystrophy, intellectual disability, developmental delay, seizures, ataxia, wide different array of diseases that affect humans. We could identify these different mutations, and about 4% of the time we would have a de novo mutation in a gene that we think is likely to be giving to us a new gene discovery. We need now to figure out what's the function of that mutation. So we've already gone through this process where, in the context of doing clinical work for diagnosing a given rare individual, we've done a new gene discovery that occurs within months rather than the years process that I described to you for the Duchenne muscular dystrophy locus years ago. So the pacing of doing gene discovery is now spectacularly faster, and we can identify many, many different rare traits. So going back to the idea of limb-girdle muscular dystrophy or limb-girdle muscular dystrophy syndromes, one of the ones that I mentioned earlier was a type of limb-girdle muscular dystrophy that looks a lot like Duchenne muscular dystrophy, which is the limb-girdle 2C, which is the gamma sarcoglycan. So it's two recessive mutations. That ends up being, most of the time, an allele that's a polymorphism in a specific population, that if you get that allele, one mutant allele from mother, one mutant allele from father, you manifest gamma sarcoglycanopathy, and it's a severe limb-girdle muscular dystrophy, much like Duchenne muscular dystrophy occurs in males and females. POMT1 is in a group of genes that affect alpha-dystroglycan glycosylation. And there's multiple different genes that those mutations lead to a very severe form of congenital muscular dystrophy and typically brain and eye malformations as well. So this is a group of diseases which are dystroglycanopathies. They're congenital muscular dystrophies, also known as Walker-Warburg syndrome would be another typical description of disease type that's caused by mutations in this group of genes. At the time when we were doing a bit of work with Kevin Campbell to do more gene discovery, there were already 6 known gene mutations that were identified in components of how sugars are put on to alpha-dystroglycan, which is outside the muscle cell membrane in the very first figure that I showed you of the dystrophin-associated glycoprotein complex. So 6 different genes, but most patients with Walker-Warburg syndrome, we couldn't find the causal mutation in. So even though we had 6 different forms, we still couldn't figure out the form for many of these. So Kevin Campbell has been working in this area trying to dissect it for years, and indeed had identified multiple independent families with evidence that they had the exact same genetic form, but that genetic form was unknown. So working with his team, where they had identified a group of patients who had a severe muscular dystrophy in the upper right-hand corner, that's a H&E section, consistent with an alpha-dystroglycan glycosylation defect, which is shown down in the lower right by immunohistochemistry and in the lower left by Western blot and glycosylation staining, as well as very severe brain malformations during development. With very clear Walker-Warburg syndrome, this ended up being one of the patients that we identified a new mutation in ISPD. It was identified partly because of linkage, like we talked about at the beginning. So 4 different families, 4 different individuals had homozygosity by descent. That is, one allele has a mutation on it, and it was the same allele in mother and father, and there was an inbreeding loop in their family, so that person manifested a rare recessive disease. And one of the families had 2 affected boys, 2 affected children, and they both happened to have 2 independent mutations that came together. So we actually ended up having 5 individuals with the linkage at the same location on a specific chromosome telling us where to look, and with combining that with whole exome sequencing, we could find multiple independent different mutations in ISPD, identifying this as the 7th known molecular cause of Walker-Warburg syndrome. However, and it actually ends up being the 2nd most common cause of Walker-Warburg syndrome genetically. So out of 100 patients roughly, 15% could be ascribed to ISPD mutations, but that still left about half of all patients that were present in the population without a mutation identified, giving a large world of new gene discovery yet to do. This is a very exciting area of science to be working in, where the tools are very powerful, and you can go about dissecting and identifying new gene discovery in a very powerful way, and then focusing on how is that mutation leading to the phenotype? How is that mutation affecting glycosylation? So already, just from studying this one rare form of limb-girdle muscular dystrophy that affects the glycosylation of one protein type, entire new sugar structures have been identified, leading to better understanding of the true biology of what the muscle looks like, but also other organ systems as well. So we have a lot of work yet to do to keep dissecting these. I like this model, and I like to show it to you, partly because it also highlights the idea that one disease, so Walker-Warburg syndrome here, is caused by, now there's over a dozen different disease genes known. This work was done a couple of years ago. But already with these modern tools, we have over a dozen different forms of, dystroglycanopathies caused by different mutations in different genes, and we still don't know some of the others. So that the genetic heterogeneity that leads to different human phenotypes is immense, and we're just now getting to explore that area. We now have new tools that we can bring to bear on dissecting what the molecular basis of a given new patient diagnosed with Duchenne muscular dystrophy in those instances where we don't find the specific mutation, and yet the child clearly has Duchenne muscular dystrophy. And this does occur. We're going to do this through whole genome DNA sequencing analysis, and that can be done from blood. And we're going to do it from RNA expression analysis, and that needs to be done from a muscle biopsy. So here's an example of that process. We've done this multiple times now, and there'll be a larger and larger fraction of these boys who we can satisfactorily find the exact mutation that caused their Duchenne. So this is a young boy who was diagnosed with Duchenne muscular dystrophy, had a clear clinical presentation completely consistent with Duchenne muscular dystrophy. Very high CK, big calves, presenting with progressive muscle weakness. Muscle biopsy was done because DNA analysis was normal for MLPA. It was normal by microarray. It's actually normal by exome sequencing as well. So he really had the battery of things done. A muscle biopsy was done, and the muscle biopsy showed dystrophic changes by the H&E, as we showed you in the earlier slide, and he had no dystrophin staining. So that's pretty consistent with the fact he's got Duchenne muscular dystrophy, and yet we can't find the mutation by sequencing the entire protein coding part of the gene, and we can't find it by very sensitive deletion duplication testing. So what gives? What could he have? So to get at that issue, we went about taking his muscle biopsy that had been done actually several years before we were working on his case, and we could take RNA from that muscle biopsy sampled years before. So it's sufficiently stable that we can do this. We take RNA from the muscle biopsy, and we just shotgun sequence all of the mRNA that's made. So every single gene is being made, every single gene is being sequenced, but here we're only suspicious of the DMD gene. So a postdoc in the lab, Richard Wang, could take all of those reads and map them back to just the DMD gene transcript, to the exon number 1 through exon number 79 that encodes the normal dystrophin gene. And what he found was exon 1 was joined to 2, et cetera, up to exon 36 is joined to exon 37, but exon 37 is never joined to exon 38. Never happened in this child, but instead, exon 37 is the last exon expressed, except for that there's a little bit of RNA expression starting at exon 63 and on. So what gives? There's clearly not a big deletion because we already did the deletion analysis, so it can't be missing. So what's going? There's not a 38 to 62 deletion here. So what gives? What gives is that through whole genome DNA sequencing with paired end sequencing reads, so you have sequences from 200 base fragments where we sequenced a little bit from the left and a little bit from the right, Richard could realign all of these data back to the genomic sequence of the dystrophin gene and the surrounding interval and identify that there were two places that were very abnormal. One, the big red sequence is a 5 million base part of the genome that got swapped and put in the wrong way, got inverted. Copy number neutral. The left end happens to be within intron 37 of the DMD gene, and the other end is 5 million bases away. So it breaks the DMD gene. It makes it impossible to make the right transcript. And indeed, you have a transcript that's made through exon 37, but no further. This is an example of what the RNA sequencing reads look like to get at that issue. What Richard was able to do was take all of the sequence data and not just say that there's this 5 million base inversion, but better than that. He could say that this 5 million base pair inversion has an exact breakpoint on the left to the base resolution and an exact breakpoint on the right to an exact base pair resolution. So we could design a very cheap DNA diagnostic where you had a PCR primer around the breakpoint on the left and a different PCR primer pair around the breakpoint on the right. And this boy is the only person on the planet who has this exact inversion. So we could see positive fragments from those two people and create an incredibly reliable, very specific DNA probe for Duchenne muscular dystrophy in this family. It'll only be relevant to this family, but it's physically possible to do. And indeed, we could use those markers to now test, is the mother a carrier? In this instance, the mother is a carrier, but then we could use that probe further to test, is her mother a carrier, which is important for her health, or her sister's a carrier, and ultimately this boy's sister needs to be tested as well. But we now have that tool in hand to be able to ask these sorts of questions and give that information back in a clinical way back to this family. So we can do this now over and over again to try to resolve these sorts of cases where we don't come up with an appropriate, full, clear molecular diagnostic. What I'll summarize here at the end is, if you think about what we're doing for Duchenne muscular dystrophy, or the very complex limb-girdle muscular dystrophies, or the specific instance of the congenital muscular dystrophies due to alpha-dystroglycan opathies, which have now a dozen different forms, it becomes very quickly apparent that we won't necessarily move forward in a way where we would identify that somebody has an alpha-dystroglycanopathy and then go about saying, is it one of these 12 different genes? And if it's not, then I'll go search for a new gene. That's really fairly old school, but instead, what we'll be doing is using these tools of diagnosis much earlier in the diagnostic workup to find the causal disease gene. So recent medical genetics practice is described in this slide, and it would typically be somebody would come into a rare disease clinic and have a phenotype, a spectrum of different issues that are wrong with them. Might be intellectual disability, might be muscle weakness, might be a very high CK, etc. And we would then take all that information, try to figure out, do they have a disease? If they had Duchenne muscular dystrophy, we can actually do that in a pretty clear-cut way because Duchenne presents in a pretty clear-cut way. And then we would only test the dystrophin gene, the DMD gene. And we're in effect using that to confirm the diagnosis at the end of the day. But consider the fact that there's thousands of different diseases like Duchenne muscular dystrophy. It's actually not practical to dissect all the phenotypes that way. And we now have fundamentally different tools where we're going to move up the genetic sequencing in the diagnostic process rather dramatically. So the average time, there's a process called the DNA, or a diagnostic odyssey, that will occur for many patients with a very rare genetic disorder. That is, Duchenne is one of the most common genetic disorders, and it takes you on average 2 years to figure it out. If you have a very rare genetic disease, it typically takes years longer than that for the physicians, their teams, and many experts to actually figure out exactly what they have. Here we now have the opportunity to invert that process, not use genetics as the last resort, but use it really early on in this process, and that's indeed what we're seeing in clinical practice. That's nicely summarized in this final slide by sort of a thought sketch by Brendan Lee, who's at Baylor College of Medicine. And this is what happens as sequencing costs decrease. So the green line is what was happening with sequencing costs using old school methods, and the red line is what actual sequence costs have done. So they plummeted rather dramatically, such that by 2012 we could launch clinical exome sequencing, and it was a meaningful diagnostic that was cost effective. the costs of phenotyping people, of determining what disease they actually have, stays stable. Those costs are staying about the same. So that at some point, and I would argue that we're already past that point, it's cheaper to do sequencing if we expect a rare genetic disease, to do that as one of the very first things that we do for kids. And then we'll be able to identify in a subset of the kids the causal mutation, and then do the appropriate targeted phenotyping in those individuals. This creates a spectacular world where gene discovery is occurring within clinical practice, and we can discover what the molecular functions are of these many different diseases, leading to developmental abnormalities, for instance, or identifying the many hundreds of yet to be discovered disease genes for muscular dystrophy, and filling out the spectrum of what human variation looks like. Thanks for your attention.